To the naked eye, the powders in these vials don't look like much. They're all white. This one is pretty fine, kind of like powdered sugar. This one is a little bit more flaky, kind of has the texture of fish food. They're not for eating though, but they are made to go in the body. Each one is a type of biomaterial that can be manipulated and sculpted to make medical devices and implants like pacemakers, knee prosthetics, and hip replacements. And in the lab of Kaitlyn Sadtler, they're being tested for their potential to help the body regenerate after a traumatic injury.
Traumatic injuries are actually quite common in the United States, about 25 million a year, and when we're looking at the more severe traumatic injuries, it's actually the leading cause of death for people under the age of 45 years old in the US.
And the people that do survive traumatic injuries often have to contend with difficult changes in the long term.
Both in terms of how those injuries look. Nobody wants to have a big scar on their face, and then also how they function. So if that big scar is compounded with an issue in using your hand, using your arm, using your leg, that extra level of difficulty that might be added to your life, it's important that we help those folks.
Dr. Sadtler is working to better understand the fundamental biology of what happens when we get hurt and how we heal. It's a tricky balancing act, especially for the immune system, which has to clear any debris and launch an attack against possible infections, while minimizing additional collateral damage and repairing the injured tissue. And as you might already know from experience, it's not always a perfect process. So how can we steady and maybe even shorten the tightrope from injury to recovery? These biomaterials could get us one step closer. Everyone can relate to being injured. Who hasn't scraped their knees in the playground or accidentally cut themselves cooking and bears the scars to prove it? To hurt is human. Most of the time, though, our injuries aren't life-threatening or life-altering, but sometimes they are.
Growing up in a spot where a lot of people were in the military, joined the military after high school. My dad was in the military, he's an ex-Marine, and he still, has a piece of shrapnel or a rock, he isn't sure which, in his lip from an injury that he got on a deployment. I knew people that got hit with grenade shrapnel and still had, scars from that and injuries from that. And, also knew people that didn't come home. And those are the sorts of people that you start wondering, how can we make that better? Both that initial survival of that injury, but then also once they come home.
But of course, major injuries extend past the line of duty into car crashes, accidents with heavy machinery, inadvertent falls, sports incidents, all of which can really happen to anyone at any time and lead to broken bones, lacerations, torn muscles or ligaments. The problem is that even with the best intentions and medical attention, our bodies can't repair themselves perfectly. The truth is, new tissue is not the same as the old.
We want to see regeneration, so growth back of tissue that's supposed to be there. So instead of scar tissue on your skin, do we get those hair follicles? Do we get freckles if you had freckles, or pigment if you had, melanin in your skin? How do we get that stuff to grow back to?
There's a long history of humanity's fascination with regeneration. It stretches back to the ancient myth Prometheus, the Greek titan who was chained to a rock and had his liver pecked out by an eagle every day, only to have it grow back at night, and continues to today with fictional characters like Wolverine, who defies death by healing instantly from any bodily harm. Centuries of scientists in between have wanted to bring fiction closer to reality, and a lot of them have looked at star healers in the animal kingdom in attempts to uncover the secrets of their regrowing abilities.
There are organisms that can regenerate, so we know it can happen. So there are things like, salamanders, they can regenerate an entire arm. Lizards can regenerate their tail. There's mice, there's mammals. The African spiny mouse can actually perfectly regenerate its skin. And so there's organisms out there that can do it. So we have our immunoengineers and then also developmental biologists that can study these different types of organisms. And throughout these kind of collaborative works between different types of science is where this field of immunoengineering really blossoms. So it's super interdisciplinary.
Immunoengineering integrates the principles of immunology with the tools of engineering to understand the mechanics of our immune system and leverage it to our advantage. By tapping into our cells and those of other animals, immunoengineers learn which molecular levers to pull and which dials to turn in order to direct how the system behaves.
One example being that salamander. There were beautiful studies early on that showed that macrophages, which is a certain type of immune cell that helps gather up clear debris and shoot off those proteins that signal to different cells, those macrophages were really important in that regeneration process. And if that salamander didn't have any macrophages, it didn't regrow its limb. And so we know the immune system's important in those organisms that can fully regenerate as well.
Us humans also have macrophages, as well as monocytes, neutrophils, B cells, T cells, the list goes on. Each has a specific job and has to be deployed at the right time after the initial moment of injury for the body to heal properly.
Imagine a motorcycle rider hits some gravel, loses grip, slides. We have all of this dirt, debris, bacteria, and an injury. Well, first of course, our immune system has to activate to prevent any sort of infection or attack any bacteria or viruses that are there to make sure that that doesn't threaten a person's life. And then after that occurs, when we've got, the wound is clotted and we're starting to heal, those immune cells can help degrade away any of the debris and the dead tissue that's left, and helps talk to different stem cells, talk to different cells that might help put down a beneficial scar. So cells like fibroblasts, all of those immune cells will talk to all of those different cells in our body. This massive interconnected process, kind of like, construction workers at a site that are talking to electricians that are talking to people that are running the plumbing, right? It takes a lot of different people to build a house. It takes a lot of different cells to build a tissue. And so the immune cells can talk to all of them and go ahead and figure out when do we want to activate certain things, because, you want your framing up before you run your electrical. And so if we can target the immune system, we might be able to start to really, pattern that response to help pattern the stem cells inside our body to build the tissue when we need it there.
So the question is, how do you build a better house? Which molecular levers do you pull and which dials do you turn to tell the immune system how to lay the groundwork for better healing? Well, that brings us back to these guys. Each material offers a set of pros and cons that make them suitable for certain applications, but not great for others. For example, polyethylene here is a synthetic plastic that can be molded with heat and cooled back down to make rigid parts. It's great for mechanical stability and weight bearing, which is good for things like knee replacements. But what if you need something softer, a little bit more flexible? For that, there's Pluronic. Another synthetic material that can be turned into a gel that is not only more pliable, but you can also load it with drugs that slowly release into the body. In the long run, though, our bodies don't generally jive with synthetic materials. We've all heard about the health risks from exposure to microplastics, how they can infiltrate all parts of the body, creating health complications and causing inflammation. The same can happen through wear and tear of implants made from these materials, as they break down into smaller particles that can't be metabolized and embed themselves in the surrounding tissue.
So how do we start moving to something that is a little bit more biologically friendly on its own, a little bit more like what our bodies are made of? So we can use different naturally dried materials. One of those is alginate. Alginate is not from animals, it's from things like seaweed. Alginate starts off as a powder, just like this Pluronic, and then we add water and it turns into a goo, just like this Pluronic. However, with this material, instead of heating it up to turn it into a gel, we go ahead and add it to salt water. So salts, like calcium, can go ahead and turn this into a gel. And so what we can do with that is implant that material. It can be used for wound healing dressings, and is a little bit more friendly to the body. Though our immune cells still can kind of tell this isn't a part of our own body. This is made from a plant. So the immune cells still look at it and go, oh, that's not me. And so how do we get closer and closer to human? We can start looking at proteins and molecules that exist within our body, such as ECM or extracellular matrix. So ECM, which is this powder here, is derived, it's basically derived from sausage casing. It's one of those things. So if you think about sausages and you take that little thin exterior off, that little piece of tissue is rich in collagen, and collagen is great for wound healing. And so what we can do is go ahead and take that same tissue. We can process it to isolate all of that natural structural material, and we can use that directly in wound healing. We can use it as powders. You can use it as that sheet, so things like a hernia repair can be done with that, all from humble sausage casing. That being said, it can be used to coat devices. So we can turn this into a gel. We can turn it into a goo that solidifies at body temperature. And those can be used and have been studied preclinically in coating synthetic materials to make the immune system respond to it a little bit better. And so what we do is we learn from what makes this good in terms of our immune response. We learn from what makes the good good, what makes the bad bad. Can we use those as design principles for that engineering art of immunoengineering? So can we leverage basically that natural fundamental background of immune responses to injury and reconstruction and implantation of materials to design new materials?
Previous research suggests that one of the bad things that can happen after traumatic injury, and could be made worse with the wrong biomaterial, is autoimmunity, which happens when the immune system mounts an attack against the body's own healthy tissue.
There is a clinical observation that in some people who undergo muscle trauma, either accidentally or through surgery, those people develop autoimmune disease.
Aditya is interested in figuring out what causes autoimmunity after injury, and what could increase the chances of developing it. He's looking for B cells, which secrete protein antibodies that act like hound dogs circulating in the blood to sniff out any intruders and signal the rest of the immune system to attack. But in an autoimmune response, B cells produce antibodies that wrongfully ID friend as foe.
Using a mouse model where I induce muscle trauma, I can detect how many of these B cells are present in these mice and how many are secreting antibodies.
To quantify B cells from a tissue sample, Aditya uses a technique called flow cytometry, which involves breaking apart a sample into individual cells and tagging them with fluorescent markers that light up in different colors depending on the type of laser that shines on them. With many different combinations of tags and lasers, a flow cytometer can distinguish between different cell types in the same sample and produce graphs that show a count of those cell populations. The researchers can then compare how the number and types of cells differ between samples to determine the immune response that corresponds with the clinical symptoms of autoimmunity.
In the animals that do have an autoimmune response, there is a type of special B cell called the plasma cell, which is responsible for producing most of the antibodies. So in figuring out which animals have these plasma cells, I can tell which animal is likely to get autoimmunity.
The presence of these plasma cells can also be used to see if and how implanting different biomaterials affect the autoimmune response. With polyethylene, for example,
we see that in mice that are healthy and do not have autoimmunity, there's little to no B cells in the muscle. But in mice that have underwent muscle trauma and have received these polyethylene implants, we see that not only do they have many B cells in the muscle, but some of those B cells become plasma cells. In mice that get a different type of implant, like extracellular matrix, we still see B cells in the muscle, but far fewer of them become plasma cells that are capable of producing antibodies. So we think these type of implants might be beneficial in terms of helping prevent autoimmunity.
The cool thing about that is we get those numbers, we get a real good view of the different immune cells that are in there, the different immune populations, and a lot more granularity. However, we've taken Humpty Dumpty apart, taken that injury space apart. We don't know where these immune cells came from, right? We lose the geography of it. And so another technique that we use in the lab is histology.
Histology keeps Humpty Dumpty more intact. In this method, the lab preserves a tissue sample in a block of wax so that it doesn't degrade over time. Then they slice it into very thin slivers, less than the 1/10 of the width of a human hair. Then, as with flow cytometry, they'll mark the sample with fluorescent tags. But in this case, all the cells have been fixed in place. So by looking at them under a microscope, they get a freeze frame of where specific types of cells lie and can map out the landscape of injury with or without a biomaterial.
And we can identify those same cells that we found with our more quantitative flow cytometry and see where those are actually sitting within our tissue. And so that gives us a better idea of, are these responding and activating because of that material that's there? Are they responding with the injured tissue? If we put in a material, do we want there to be a modified coating on the edge? Is that going to be where these immune cells are going to be recruited? Or do we want it to be something that's all throughout the material? And so what we do is we go ahead and we basically identify levers to pull. And that is something in terms of therapeutics. If you have a target, you can say, I want to activate that. That's a target for a drug. That's a target for a new material. And so we identify those targets. We have other projects going on in the lab where previously we've identified immune cells that help this material do what it does. And now we're trying to use those kind of natural stimuli to promote that activity and promote regeneration.
Because that's what it all boils down to. How we can harness the power of biology to develop new strategies for therapeutics, to rebuild cell by cell and repair damage once considered irreversible, so that recovery doesn't just mean mending, but truly regenerating.
Pie in the sky dreams, of course, are, if a salamander can do it, why can't we regenerate an arm, right? Why can't we regenerate an organ? And you talk to anyone in the field, it's somewhere they want to see the field going. And I certainly believe that in the future that'll happen. I say it's not going to happen next week. It's, it's some effort to get things moving. We know it's possible. And I think that that's the exciting part of it. So, downstream some of these science fiction things starting to become reality. But, even in the short term, we're talking about, people that want to make their scars better. If somebody who's got an injury to their arm is able to open up a cabinet that they couldn't open up before, somebody's able to stand up even just for a little bit, and they weren't able to stand up before. These incremental advances, it's not necessarily just one giant leap forward from nothing to regenerating armor. So there's a lot of things we can do along that way that really help people's quality of life.
For more stories like this that take you inside the labs of researchers at NIH, check out our website at irp.nih.gov and follow us on our socials linked in the description below.